The large step prior to mitosis is called interphase. While prokaryotic cells like bacteria replicate through binary fission, eukaryotic cells divide through mitosis. All eukaryotic cells use mitosis as a means of duplicating cells. Interphase is an integral part of the cell cycle that prepares a cell by producing the proteins and duplicating chromosomes. Interphase was once considered the resting stage of the cell cycle because microscopes could not detect movement in the cells. However, it is now known that interphase is a period of 3 of the following stages. During G1, or gap stage, cells grow and synthesize proteins needed for mitosis. These proteins also activate the cell's metabolism and break down carbohydrates and lipids to transform food into energy. Chromosomes inside the cell have not yet replicated in G1, so each chromosome contains only one DNA molecule. The S phase follows G1, and its major function involves replicating the cell's DNA. The S name stands for synthesis, because through using enzymes, the cell copies the DNA molecules of each chromosome. After the S phase completes, chromosomes have 2 chromatids, meaning each chromosome has 2 molecules of DNA. After the S phase, a cell enters G2, or gap 2, which involves another period of growth. The cell synthesizes more proteins and organelles. The cell also resumes normal functions like transforming food into fuel. Cells also stock up on this energy for its use in mitosis. G2 is the final step before a cell enters the first stage of mitosis.
Prophase is the first official stage in mitosis. Chromosomes become visible, the nucleolus disappears, the mitotic spindle forms, and the nuclear envelope disappears. Each duplicated chromosome is seen as a pair of sister chromatids joined by a duplicated but unseparated centromere. Chromosomes also become more coiled and can be viewed under a light microscope. The nucleolus also disappears during prophase, and the mitotic spindle, consisting of microtubules and other proteins, form 2 pairs of centrioles as they migrate to opposite poles of the cell. The nuclear envelope then disappears towards the end of prophase. This signals the very beginning of the metaphase.
Metaphase is the second official stage in mitosis. The chromosomes become arranged on the metaphase plate and they are attached to the now fully formed spindle. The centrosomes are now at opposite poles of the cell. While the chromosomes are now highly coiled and condensed, they become arranged on a plane that is equidistant from the 2 poles called the metaphase plate. For each chromosome, the kinetochores of the sister chromatids face the opposite poles, and each is attached to a kinetochore microtubule coming from that pole. The cell is now ready for anaphase. Anaphase is the third stage of mitosis. Anaphase begins when the duplicated centromeres of each pair of sister chromatids separate, and now the daughter chromosomes begin moving toward opposite poles of the cell due to the action of the spindle. Depending where the centromere is located along the chromosome, a characteristic shape appears during chromosome movement at the end of anaphase. A complete set of chromosomes has assembled at each pole of the cell, signaling the start of telophase.
Telophase is the fourth and final stage of mitosis. The chromosomes assemble in sets at the 2 poles and begin to uncoil, eventually assuming the extended state characteristic of interphase. A nuclear envelope reforms around each chromosome set, the spindle disappears, and the nucleolus reforms. Nuclear division by mitosis is complete at this point, and cytokinesis, the division of cytoplasm, has already begun. In animal cells, cytokinesis involves the formation of the cleavage furrow, resulting in the pinching of the cell into 2. In some other specialized cases, such as in the development of the Drosophila embryo, nuclear division occurs without being followed by cytokinesis. This completes the cell cycle.